Greetings, members of the jury, and to everyone else watching this presentation. Let me introduce myself. My name is Alfredo 𝐘áñ𝐞𝐳𝐶, and I am a student of the Facultad de Ciencias Químicas of the Benemérita Universidad Autónoma de Puebla. I am going to present you the poster under the title Hippocampal Ethanol Toxicity and Long Term Potentiation Impairment. First, as an introduction, we know that the hippocampus is one of the most studied brain structures involved in complex cognition processes. It is located under the cerebral cortex and contains two main regions, the cornua monis and the dentate gyrus, shown in figure number 1. Here in this figure, we show a kernel slice of the hippocampus. The most prominent functions of the hippocampus are, first, regulation of emotions, second, spatial memory processing, and third, learning and memory. The objective of this poster is to review ethanol effects on brain cells to approach a better understanding of its toxicity. Talking about long-term potentiation, it is generally divided into two distinguishable phases. The first is an early phase, which is independent of protein synthesis, ELTP, and it is shown in the figure number 2. This process is mainly led by calcium MK2 kinase, and it's going to produce AMPA receptor phosphorylation and AMPA receptor externalization. The second is a late phase, which involves a variety of intracellular signaling pathways and deactivation of transcription factors, making this stage dependent on protein synthesis, and it's shown in figure number 3. Here in this figure, the two main mechanisms leading to LLTP is calcium MK2 signaling and BDNF signaling, or brain-derived neurotrophic factor. Both pathways are going to produce a phosphorylation of CREB, thus giving a rise to protein synthesis. Now, talking properly about ethanol toxicity, the processes by which ethanol exerts its toxicity depends mainly by two factors. The first is the stage of development being administered, and the second is the exposure model. The mechanisms leading to neurotoxicity are broad, from hindering LTP to postsynaptic cell damage. These are summarized in the following figure, figure number 4. The first mechanism is GABA-A receptor agonism, thus having a diminishment on glutamate release onto the synapse. The second is GABA-A receptor downregulation. The third is NMDA receptor blockage. And the fourth is mitochondrial damage. This might be further divided into 4 main processes. The first is oxidative stress, leading to cell damage. The second is impaired mitochondrial dynamics. The third is a decrease on mitochondrial permeability transition pore expression. And the fourth is a diminishment on the expression of oxidative phosphorylation complexes. Finally, the conclusion is that ethanol has been acknowledged for causing cognitive impairment in humans and animal models, with its effects taking up to 8 weeks to completely restore normal cellular function. Taking attention to ethanol mechanisms of toxicity is relevant due to its high availability and consumption among the young and adults. Understanding the bench and chronic exposures is vital because these are the main drinking behaviors in these populations. Thank you.